Hello viewers. While talking about genome mapping, we saw that we need certain markers. Initially, they used to be the gene markers, then came the biochemical markers, but ultimately nowadays molecular markers they are being used in order to map the or locate the position of a particular gene in the genome. So in this video of ours, we are going to talk about a molecular marker that is RFLP. RFLP or the restriction fragment length polymorphism. This technique, it basically depends upon the variation in the homologous DNA sequences. Variation in the length of the fragments that are obtained when this particular homologous DNA sequence is subjected to a restriction endonuclease. That is, when treated with a restriction endonuclease, a given sequence of DNA, it may yield different length fragments. Some fragments may be smaller as compared to other, depending upon the position and number of the recognition sequence for that particular restriction endonuclease. And this polymorphism is made use of as the molecular marker. Now, this technique in this way, it is based on or it uses the activity of restriction endonuclease enzyme in order to detect the variations in the given DNA segment. So for this technique, we subject the genomic DNA to a particular restriction enzyme and we obtain different fragments of the genomic DNA. Because this restriction endonuclease, it will break the phosphodiester bonds and at the particular recognition sequences, and we have talked about these restriction enzymes in another video of ours, and the fragments thus obtained, they are of different lengths, so they are separated in accordance with their length by means of gel electrophoresis. Now, once these DNA fragments they are present on the gel, they are denatured by using some alkali. Denaturing of DNA means we are separating the two strands of the DNA molecule and by breaking up the hydrogen bonds by changing the pH of this particular solution in which it is placed, this gel is placed. So now this gel of ours, it will be having the single stranded DNAs, and these single stranded DNAs, they are then transferred from this gel to a membrane, and this is done by another technique, that is the southern hybridization technique. In this method, uh, this membrane, it is placed under weight, and this DNA, single stranded DNAs, they are transferred on this membrane because of this weight that is present on this gel and membrane combination. So by southern hybridization, we transfer the single stranded DNA to the membrane, and then after this single stranded DNA, we have to recognize or we have to detect the presence of a desired sequence of nucleotides. So that is done by making use of specific RFLP probes. So they are a probe, that means they are labeled DNA sequences, and this labeling, it may be of some fluorescent molecule, or it may be the radioactive, that is P32 can be the label that is used for this RFLP probe. So this single-stranded stretch of DNA, it will hybridize with its complementary sequence that is present in the stretch of fragments that we obtain that are present on the membrane now. These probes which are used, they are typically the short or the single or low copy genomic DNA, or they may be the complementary DNA clones. These probes, they are used in the genome mapping as well as in the variation analysis techniques like the genotyping or the forensic studies in order to identify the given stretch of DNA, or in the paternity tests, or the to for the diagnosis of hereditary disease, etc. So these RFLP probes, they are incubated with the membrane on which now we have got a single stranded DNA. So when they are incubated together, these probes, they will hybridize, they will form double stranded stretches to their complementary strand on this DNA present on this membrane. And then after, these probes, they are transferred to an X-ray film. So these probes, they have got this marker part, and once they are transferred to the X-ray film, we can easily view the position of these fragments that contain a desired oligonucleotide sequence. So our DNA stretch, it has got the complementary sequence to the probe, and along with these probe binding sequences, it also has got the recognition sequence for some particular restriction endonuclease enzyme. And all these three samples, if we consider, all of them, they have got the similar probe binding sequence. But there's one difference, that in this sample B, because of some mutation, this recognition sequence which is present at this point in sample A and C, it is not there. That means this sample B has got just two recognition sequences at which the phosphodiester bond, it may be broken up by the given restriction endonuclease enzyme. So when the sample A is subjected to restriction endonuclease, these are the three recognition sites at which the phosphodiester bond, it will break, and it will lead to the formation of two types of fragments. One at which the probe will hybridize and it can be detected, and the other in which no recognition sequence is there. So the probe, it will not bind to the sequence and we won't be able to detect it on the X-ray film. Now similarly, if we digest the sample B with the same restriction endonuclease, again we will get the fragments, but this time, since this particular recognition sequence is not there because of some mutation, this sequence, it has altered. So now we have got this one particular segment of DNA which has got the probe binding sequence, but its length is larger as compared to the one that we obtained in case of sample A. So on this X-ray film, when we'll run the gel in gel electrophoresis, at that point of time, this particular fragment of DNA, it will travel less, because we know that in gel electrophoresis, the smaller the DNA stretches which are smaller, they move faster as compared to the ones which are longer. So this is the band of sample B that has got the probe attached to it. And now in sample C, there's no mutation at this second point, this point number 2. So again, similar pattern of this gel electrophoresis will be obtained and observed on the X-ray film. So we can use this type of segregation for analysis of inheritance, because we know that in a gel on which the this solution is run, in case of gel electrophoresis, the smaller DNA segments, they will be towards the lower end, and the larger DNA segments, they will be towards the upper end of the gel. Now, just now we have seen that if some site of recognition site, it undergoes mutation, it leads to the formation of larger fragments. So in this case, this homozygous dominant condition of A is the mutated uh, DNA fragment, whereas the wild type is the homozygous, this recessive or the small a. And in case of heterozygous sample, we will obtain this probe. It will attach at both the sides. That is, we will obtain fragments of DNA of both lengths. So we will know that they are the points of homologous chromosomes, and this is the homozygous chromosome. And when heterozygous chromosome is there, or heterozygous alleles they are present in the given sample, at that point of time, uh, instead of one, two bands will be obtained on the X-ray film. So in this manner, we can analyze not only the inheritance pattern, but also the mutations which destroy one of the restriction sites by using this method. So these RFLPs as molecular marker, it is very specific to a particular given single clone and restriction endonuclease combination. And most of the RFLP markers, they are codominant. That is, the two alleles in the heterozygous sample, they will be detected because they are in the form of single stranded DNAs. And the probe, it will bind to both the stretches of wherever it has got its complementary restriction site. And because of this uh, complementary restriction site, these RFLP molecular markers, they are highly locus specific. So we can identify particular stretches of nucleotide sequences or oligonucleotides in a given gene or genome, and in this manner we can use these RFLPs as molecular markers even for gene mapping. We will talk about other molecular markers in the videos that will follow.